Good morning, everybody. Today, I'll be discussing pathophysiology of sarcopenia and cirrhosis, and I'll be sharing some published and unpublished data in this area. As we all know that sarcopenia is loss of muscle mass and muscle strength, which is associated with aging and chronic illness. It is shown to be an important factor in determining quality of life and long-term outcome in patients with cirrhosis. So, for studying the mechanism, metabolic tracer kinetic studies and molecular signaling pathways were studied in sarcopenia. We know that in cirrhosis there is significant malnutrition, maybe due to inadequate intake or malabsorption and maldigestion. This is associated with systemic inflammation, metabolic dysregulation, visceral fat accumulation, insulin resistance, and endocrine dysfunction. There may be physical inactivity. This may lead to a clinical phenotype of frailty and sarcopenia, which is associated with decreased health-related quality of life. And why this is important to study loss of muscle in liver disease? Because muscle loss is the most common complication in cirrhosis. And what are the consequences of sarcopenia? There is increased risk of hepatic encephalopathy, increased risk of infection and sepsis, hepatocellular carcinoma, negatively impact the quality of life, poor outcome of liver transplant. There is increased hospitalization, prolonged ICU stay, longer duration of intubation, increased hospital cost, and increased waitlist mortality. So prevalence varies, and if we apply different criteria, it varies from 52 to 35%, and if we apply Asian criteria, it is 19% in males and 20% in female, according to our own data. So what are the factors which are responsible for cirrhosis, for sarcopenia and cirrhosis? That is important one is increased dietary intake, decreased testosterone level, decreased growth hormones, inflammation, decreased insulin-like growth factor 1, hyperammonemia, inactivity, and altered gut microbiome. The most studied is in cirrhosis, hyperammonemia. So what are the causes of decreased food intake? There may be nausea, early satiety due to ascites. There may be upregulation of leptin and TNF-alpha, which can lead to decreased appetite. There may be altered taste due to zinc deficiency, and food may also not look very tasty because we generally advise salt restriction, and they are fasting for clinical investigations, and there may be an element of encephalopathy as well. So what happens in cirrhosis actually? It's a state of accelerated starvation. So the changes in metabolism which we see after 3 days of fasting in a normal individual, that occurs after overnight fast in patients with cirrhosis. So provision of adequate calorie prevent fasting and skipping meals. And what is the dynamics of fuel metabolism? Patients with cirrhosis have significantly decreased carbohydrate oxidation and increased fat oxidation. So the pattern of fuel use in patients with cirrhosis resembles that in starvation, and this is what we have observed, that the substrate utilization is altered in chronic liver disease. The RQ is significantly lower after overnight fast. In the morning it was around 0.75 plus minus 0.03. That shows that the major substrate which is oxidized is fat, not the carbohydrate. The resting energy expenditure is significantly raised in patients with CLD compared to healthy individual. So in nutshell, what happens? There is a state of accelerated starvation, reduced glycogen store, increased gluconeogenesis, especially from the breakdown of the protein, increased fatty acid oxidation, beta oxidation, and gluconeogenesis from amino acids and not from fatty acids. This and the only source of amino acid is proteolysis of muscle that generate branched chain amino acid and aromatic amino acids. A skeletal muscle can utilize only branched chain amino acids due to localization of branched chain keto dehydrogenase is there, and so aromatic amino acid utilized only in liver, and low branched chain amino acid and high aromatic amino acid concentration in plasma. What happens to the liver cell? We know in cirrhosis the hepatic dysfunction is there, And hepatic synthesis of glucose from alternate substances like and glucogenic amino acids, especially glutamine and alanine from the skeletal muscle. There is temporary glycogen stores. There is depletion of glycogen, which is there in the liver, and destruction of liver architecture in cirrhosis. There is persistent compromise of function and decline in the activity of glucokinase. This is the enzyme which in the liver that is responsible for phosphorylating glucose as the first step in hepatic glycolysis, and increased reliance on gluconeogenesis due to protein catabolism. So what is the mechanism actually in skeletal if skeletal muscle mass is there, there is protein synthesis and protein breakdown. This process goes on continuously. There are satellite cells which proliferate and differentiate into myocytes, and myostatin actually downregulate protein synthesis, and insulin-like growth factor 1 is stimulate protein synthesis. There are two pathways, ubiquitin proteasome pathway and lysosomal autophagy pathway, which are responsible for degradation of protein. So what are the potential mediators of liver muscle axis? The proposed potential mediators are ammonia, most studied, decreased testosterone and growth hormones, endotoxemia, altered immunity and gut permeability. Even though there is evidence to support each of these potential mediators, hyperammonia has been studied most extensively in chronic liver disease. What happens where this ammonia is produced? As we all know, that the colonic flora that generate ammonia, this reaches to the liver, and in liver, through urea cycle, this is converted into urea and excreted through the kidney. Because hepatic function is compromised to some extent, so urea cycle is also impaired, and there are shunts. So ammonia can reach directly to the circulation to the brain and may lead to encephalopathy, and in muscle, it is converted into glutamine. This is a study in which they have shown that the plasma and muscle level in cirrhosis, the ammonia level is raised, and they created two models. So once there is shunting and hepatic dysfunction, hyperammonemia means there is more ammonia which is available in the muscles, and then it through NF-kappa B pathway, it increased the expression of myostatin, because this along with NF-kappa B, it upregulate myostatin, which decrease protein synthesis, and through SMET2 and 3, it also upregulate ertogen, which is an autophagy gene, and MRF, which is responsible for ubiquitin proteasome pathway. In mitochondria, that also causes injury due to Krebs cycle intermediates are not available, so energy production means ATP generation is less, and there may be mitochondrial injury, and that will lead to reactive oxygen species generation, and that also inhibit mTOR pathway. This is how ammonia act in the cytoplasm. This complex P65 inhibitory kappa B complex, it releases inflammatory kappa B, and P65 enter into the nucleus, and where with NF-kappa B, it upregulates myostatin expression. This is very important. In fact, in citric acid cycle, what happens? The acetyl-CoA, pyruvate to acetyl-CoA, this acetyl-CoA is not available as an intermediary, and same way alpha-ketoglutarate, that in high, if ammonia is high, then the Km value from alpha-ketoglutarate to glutamate is very high. So only if ammonia level is raised, alpha-ketoglutarate will go into this direction, that is glutamine and glutamate. And so if it is not entering into citric acid cycle, then the ATP generation will be impaired. When there is hyperammonemia, along with myostatin, it also means GCN2 is a sensor which senses for the deficiency of amino acids, and then it leads to eukaryotic elongation factor 2 alpha. It phosphorylates that, and that also inhibits protein synthesis to some extent. But what happens in this process, the glutamine which is secreted, which is synthesized in the cytoplasm, it goes into the circulation, and through a transporter, leucine enter inside the cell, which is utilized for the synthesis of protein. So circulating level of leucine may be low, but inside the cell, leucine is available for muscle synthesis. This is how what happens if there is high ammonia. It lead to stimulation of HIF1 alpha. If once this is stabilized, it will upregulate or it will stimulate PDK means, and this PDK with pyruvate dehydrogenase, it inhibits pyruvate dehydrogenase. Pyruvate will not be available, and so pyruvate will not enter through acetyl-CoA in cycle. And same way, it also inhibit alpha ketoglutarate going into the citric acid cycle, which will lead to decreased ATP generation. Proteasome activity is unaltered in cirrhotic skeletal muscle. This is seen in hyperammonemia mediated autophagy in skeletal muscle contribute to the sarcopenia of cirrhosis. As far as autophagy genes are concerned, bacillin and LC31 is upregulated. Then there are two co-transporter. This has been shown in a recent paper in a knockout mice model of MYD88 and RHBG positive knockout mouse model. They have given ammonia through an infusion pump for a month in a limited quantity, and then saw the expression of various protein, and they have shown that this co-transporter is required for NF-kappa B functioning in the cells. This is also what we have seen, that senescence associated molecules P21 and P53, they are upregulated in patients with cirrhosis having sarcopenia. Then in myotubules, what we have observed, that the ATP content especially in sarcopenic patients were less, and when they were treated with ammonia, and there was change in the size of myotubules, and if we give flavonoids like camphorol, this change could be reversed to some extent. This is our own data, and where we have in 180 cases we have seen that the prevalence of sarcopenia is around 15%. Then what we have observed, that myostatin level is raised in patients with cirrhosis in the blood, and especially if patients having advanced disease, more in B and C. There was no correlation between ammonia and myostatin level. And then to study the mechanism, you see that there is decreased protein synthesis and increased protein breakdown. So we studied MSTN, AKT, mTOR pathway and gene expression of MSTN, ISC for AKT and mTOR. For autophagy pathway, we look for gene expression of bacillin and LC3. For apoptosis, annexin B and caspase 3. And for ubiquitin proteasome pathway, gene expression of TRIM63 and FBXO32. This was assessed in 10 patients, and what we have observed, that mTOR signaling in sarcopenia was impaired. Same is true for AKT also, and autophagy and this transcription level of MSTN, BCN, and LC3 was also impaired. Especially MSTN was more expressed in sarcopenic patients. So there is increase in autophagy and myostatin gene expression and impaired mTOR signaling in patients with sarcopenia and cirrhosis. What happened again in the muscle, this is on histochemistry, that there was increase in angulated fibers suggesting muscle atrophy and deposition of fat in muscles. The same thing here also. And this is the transmission electron microscopy showing changes in disintegrated sarcolemma is seen a structure with increased dissolution of Z and M line protein, altered mitochondrial morphology, which are swollen, and muscle glycogen is decreased. This is again transcription level of MSTN, which is increased in cirrhosis, same slide, and MRI expression of bacillin 1 and LC3 was increased, and as far as apoptosis pathway is concerned, there is no change. So in summary, what we can say, that there is upregulated MSTN, downregulated mTOR, and upregulated bacillin. So autophagy is upregulated, and there is no change in ubiquitin proteasome pathway. Now coming to vitamin D, that also play an important role because it leads to upregulation of insulin-like growth factor, IGF-1 and follistatin. Follistatin is an antagonist to myostatin. So it promotes cell differentiation and proliferation. And at the mitochondrial level, again, if there is deficiency of vitamin D, then there is increased ROS production, increased oxidative damage and muscle atrophy. And when we give vitamin D through vitamin D receptors, these changes can be reversed to some extent. What we have observed that there is deficiency of vitamin D in patients with cirrhosis, and the sarcopenia was present in 36.6% of cases and osteoporosis in 37.3%. Only and prevalence is high, especially in child C cirrhosis. Then coming to role of hormones, as we have seen, the ghrelin increases appetite and leptin decreases appetite. So leptin is raised in patients with cirrhosis. Then there are growth hormones which are decreased in CLD, especially testosterone. Testosterone act through PI3K and AKT pathway, AKT-mTOR pathway, which is pathway for muscle synthesis. So if there is deficiency of testosterone, there will be diminished mTOR activity leading to decreased protein synthesis. Same way IGF-1, insulin-like growth factor, also stimulate the same pathway. So there is deficiency of insulin, this factor also. There is insulin resistance, which lead to decreased hepatic clearance, increased shunting and pancreatic secretion. This can also lead to protein breakdown. This is again representation. Testosterone again acts through AKT-mTOR pathway, leading to synthesis and presence of amino acids. And same way insulin-like growth factor also act through AKT-mTOR pathway. Vitamin D through follistatin. Follistatin, by inhibiting effect of myostatin, will increase the AKT-mTOR pathway expression and synthesis of proteins. Same way ammonia I have already discussed. Then coming to the role of cytokine, there is immune dysfunction and there is increased oxidative stress and bacterial migration from the gut, because gut is leaky in cirrhosis, and there is transmigration of gut bacteria products, PAMP and DAMP, and that lead to inflammation in the liver, because Kupffer cells, they will recognize these toll-like receptors are there, and they will lead to expression of various cytokines, especially TNF-alpha and interleukin-1, which act through NF-kappa B and MRF, leading to upregulation of ubiquitin proteasome pathway and proteolysis. Now coming to the role of portal hypertension. What happens in portal hypertension? There is splanchnic vasodilatation, and so there is activation of RAS system. This stimulates salt and water reabsorption. And what effect this has on muscle? It stimulates activity of NADPH oxidase and lead to reactive oxygen species generation. It also increases ertogen-1 and MRF1, leading to overexpression of ubiquitin proteasome complex, and it increases cortisol level that also lead to sarcopenia or muscle destruction. So angiotensin II, this disrupt binding of insulin-like growth factor, because these receptors, they occupy. So the activity of insulin-like growth factor is impaired, and that same way activity of mTOR and AKT pathway will also be impaired when this is there, which will lead to muscle loss. Now coming to environmental factor and gut dysbiosis, we all know that there is dysbiosis in the cirrhosis. There is decreased bacterial diversity, there is decreased short chain fatty acid production, increased intestinal permeability, increased bacterial translocation, decreased antioxidant function, and endotoxins, and SIBO also. This all can contribute to sarcopenia. This is again the same thing. This is our own data where we have shown that both in NFLD and ALD, gut microbiome found to be altered and reduced alpha diversity and Firmicutes bacterite ratio. Then when we did lactulose mannitol test, that permeability is also altered, and there is change in TJP, means junctional protein expression, and endotoxemia was also high in cirrhosis. So systemic inflammation and endotoxemia are elevated in cirrhosis and further rise during varicose bleed. This we have shown in 120 patients after varicose bleed. There was a significant rise. Now coming to the physical activity and sarcopenia, as we all know that due to once we indulge in physical activity, the insulin-like growth factor is increased, which will stimulate AKT pathway and will lead to muscle synthesis. And if we are not working, this will lead to increased myostatin level and myostatin expression through FOXO protein expression that will also stimulate ubiquitin proteasome pathway, which will lead to proteolysis. And if we are not exercising, then testosterone and insulin-like growth factor and growth factors are also less, so mTOR pathway will also be inhibited. That will lead to sarcopenia. There are certain myokines. I will not go into the details. These are muscle-secreted small proteins. They have an anti-inflammatory immunoprotective effect, and there are more than 100 myokines which are recognized. There are positive regulators like follistatin and BMP, that is bone morphogenic protein, and irisin. Irisin is increased during exercise, and there are certain negative regulators like myostatin, TGF-beta, activin, and GDF11. This is our own data where we have clearly shown that in cirrhosis, especially those with sarcopenia and without, there was a significant difference in the interferon gamma, interleukin 18, activin, myostatin, interleukin 6, and interleukin 1 receptor antagonist. And so in nutshell, we can say that portal hypertension, pro-inflammatory cytokine, hyperammonemia, hyperaldosteronemia, glucocorticoids, and impaired insulin-like growth factor signaling, alcohol intake, and inactivity will lead to sarcopenia. So in summary, we can say that sarcopenia is a frequent complication that adversely affects clinical outcome. Alteration in food intake, hypermetabolism, alteration in amino acid profile, endotoxemia, accelerated starvation, and decreased motility may all contribute to sarcopenia and cirrhosis. Hyperammonemia gained attention as a possible mediator of the liver-muscle axis. Increased muscle ammonia causes catapodosis of alpha-ketoglutarate, transport of leucine in exchange of glutamine, impaired signaling by leucine, expression of myosin, which is upregulated, phosphorylation of eukaryotic initiation factor 2, mitochondrial dysfunction, increased ROS, and decreased protein synthesis and autophagy-mediated proteolysis also play a role, and these molecular and metabolic alteration may contribute to the anabolic resistance and inadequate response to nutrient supplementation in cirrhosis. Thank you.